Hello. In this video we're thinking about fermenters and we're thinking about yogurt making. We're thinking about this for IGCSE biology, and specifically these are paper 2 topics, both of them. This is an image of a fermenter and there are various parts of it, and we're going to think about what those parts do. This in here is the medium of the fermenter, and in here you will have microbes, you will have nutrients in, and you'll have air going in. In particular, you'll need O2 going in most cases. The microbes that go in here are going to be ones that you consider particularly valuable. You want to grow lots of them. Maybe, for example, they are transgenic bacteria to make human insulin, in which case we're going to need it to be aerobic, and we're going to want to have lots of glucose in here, and we're going to have lots of amino acids in here. You're going to need these and other nutrients besides, because you want glucose for your bacteria to have fuel for respiration, and you want amino acids so that they can make proteins and grow, but also so that they can make human insulin itself. That itself is a protein, and amino acids are the building blocks of proteins. All this around here is a fluid, is a liquid suspension and solution of these various things. Now, of course, microbes themselves don't dissolve in water, and so given the opportunity, they will just settle out to the bottom down here. And that's no good at all, because the ones on the top, well, maybe they'd be all right, but the ones in the middle, well, they wouldn't get any amino acids or glucose or indeed oxygen, which is going to be dissolved in this water, and you'd also get a build-up of carbon dioxide in the middle of those piles. So we don't want settling. Settling is no good for us, and that's why we have these paddle stirrers here to keep it going around, to keep it well oxygenated and to stop CO2 from building up, and to make sure all microbes get a fair supply of food. CO2 itself will of course be made, and so that CO2 will come out here, and we have our gas outlet to allow that to come out. So we've got O2 going in, we've got CO2 coming out. In fact, we kind of need to do the gas exchange for the microbes in this way. How do we know when to do that? Well, we have these probes. They will detect levels of CO2 and of O2, and that will tell the computer what it needs to do. That CO2, of course, is produced by aerobic respiration. Aerobic respiration also generates heat energy. Now, we want the fermenter to be at a reasonable temperature in order for the bacterial enzymes to be working at their optimum temperature. But we don't want it to get too hot, and therefore typically actually we have cooling water going in. This is a cooling jacket here, and it will come out slightly warmer. It will be taking heat energy away, and so therefore it will cool the fermenter, keeping it at an optimum temperature, again as controlled by the computer. We want a tap for the product, and then you'll need to purify your product later on in downstream processing. And there's one little thing that maybe we've missed out on this diagram, and that would be here. That would be a filter for the air going in. It's going to be really important that we stop other microbes getting in. We have made in this fermenter a lovely, warm, gooey, salty, sugary, amino acidy, oxygenated solution, which is perfect for microbes to grow. Now, of course, that's not just perfect for our transgenic bacteria to grow as we want them to, but it's also perfect for any other microbe to grow. And so we got to make sure those things don't get in there. So we filter for the air in, and that will keep the conditions sterile. That's really important. And of course, beforehand, you're going to want to clean it properly so that there aren't any bacteria in there before you start, and you do that with superheated steam. Now, there's a good reason to use superheated steam rather than bleach or something like that, which will of course sterilize it, but you don't want bleach in your product. Superheated steam, on the other hand, will cover every single surface in that fermenter and will then just condense out to water, which is perfect. It leaves no toxic residue behind. Thank you, superheated steam. Transgenic bacteria aren't the only things you might want to grow in a fermenter. There are two other things that you need to think about for GCSE. That is yeast in brewing. Now, that's a paper 1 topic. We're not going to cover that in this video. but also bacteria for yogurt making. And that we're just about to have a little look at. For both yeast in brewing and yogurt making for bacteria, we need anaerobic conditions because we're interested in the product of anaerobic respiration in those cases. Yeast in brewing, we want the ethanol that's made by yeast when they respire anaerobically. That's going to be the alcohol in fermented drinks, such as beer. And in yogurt making, we want the lactic acid made by those bacteria, and we'll have a look at why in just a moment. This is a summary of what we've just talked about for the fermenters. You might want to pause on this bit and jot it down if that's going to be useful for you. And I'm going to carry on. Here we go. Yogurt, making yogurt. This here is a yogurt maker. It's pretty simple. You can buy them off an internet supplier. And in order to make your yogurt, you get UHT milk. It's important that it's UHT. I'll talk about that in a moment. And you pour that in, and then you take 1 spoonful of live culture yogurt. Now, the live culture yogurt contains this bacterium here, Lactobacillus bulgaricus. Now, it may contain other bacteria as well, but this is the one that's specifically mentioned in the IGCSE syllabus. And this bacterium is still alive in this culture. We just need 1 spoonful. So 1 spoonful, out it comes, thank you, and in you go. And that will be enough. The bacteria alive in that spoonful of yogurt will now be able to feed on this UHT milk and get all its lovely proteins and sugars and fats and themselves reproduce. Okay, why does this make yogurt? Well, when we incubate this at 37 degrees C, our Lactobacillus bulgaricus, their enzymes will work at optimum temperature and they will do this conversion. They will take the sugar in milk, lactose, and they will convert it into lactic acid via a number of steps, but essentially we can just remember it as being by anaerobic respiration. Now that's just the same sort of anaerobic respiration that you learned about in muscle cells producing lactic acid. That lactic acid will have the effect of lowering the pH, and that will have 2 different effects. Number 1, it will preserve the yogurt for longer. Now that's because the other bacteria who might want to come and live in this yogurt, they don't like this acidic pH. And so your yogurt will last a lot longer in the fridge than your milk will. In fact, your milk will turn to something not dissimilar to yogurt if you leave it there. It gets all clumpy and lumpy and no one likes it. No one likes that. And it has the other effect of denaturing the milk proteins. Now, normally you think about that as a bad thing, but what that actually happens, it causes those soluble milk proteins. Of course, milk, it's got to contain protein because it's got to feed a baby mammal, and it's soluble, but once you expose it to acid, those proteins coagulate, they solidify. Now, once those proteins coagulate, that thickens the yogurt. And that's what gives you your texture of the yogurt that you're probably very familiar with, and maybe you love it, or maybe you, like me, you think it's kind of, it's all right. It's not great, it's all right. But there we go, and that's how you make it. We mentioned there was a reason that you use UHT milk. UHT milk has been treated at a really high temperature, an ultra high temperature. That's in order to totally sterilize it. That will give it a much longer shelf life, of course, but also from our point of view, it'll mean that we start off with no other bacteria in our yogurt maker, in our milk, and we've got unfettered access to all those nutrients for Lactobacillus bulgaricus. It suffers no competition from other microbes who might come along and do things we don't want them to do to our milk and make our yogurt all horrible. Well, that's yogurt making. I hope that's a helpful video. Do like and subscribe if that's the kind of thing you do. Thank you.